Good morning, everybody. Uh, we welcome to the online classes on gastrointestinal system. This is a new chapter over which you will be encompassing the normal digestion absorption for which you need 5 secretions, mainly saliva, gastric juice, bile, succus centricus, and pancreatic juice, with some movements which is called as gastrointestinal motility. Following which you should know the absorption in the small intestine, predominantly the duodenum and the ileum, and the motility which happens in the large intestine, which involves defecation. The main motility is in the upper GI is the deglutition, which is the swallowing after you chew the food. Let's come across the functions of the GIT. The function of the gastrointestinal tract is to provide nutrient, water, electrolytes, vitamins, and minerals. Before this, food must be moved along the alimentary tract for digestion and absorption. The functions of gastrointestinal tract may be considered as the movement of food through the gastrointestinal tract, secretion of digestive juices, that is saliva, gastric juice, small intestinal juice called the succus centricus, pancreatic juice, and the liver secretion, which is the bile, and finally the absorption of products of digestion and water, electrolytes, and vitamins. An overview of the intestinal function is saliva secreted in the mouth, which enhances the digestion of the cooked starch. Stomach, wherein the digestion of protein starts with the involvement of pepsin, which converts pepsinogen to pepsin in the presence of hydrochloric acid, and duodenum, which secretes bicarbonate and mucus from the Brunner's glands, and which enhances digestion of carbohydrates, proteins, and fat by enzymes of pancreatic juice. Pancreatic juice also, in duodenum there is absorption of iron, calcium, copper, fat, and soluble vitamins. In jejunum, which is nothing but empty, helps in digestion and absorption of end products of digestion and absorption of water. Ileum, digestion further continues, and there is absorption of the following above products. And there is also absorption of vitamin B12 and bile salts in the presence of vitamin B12, which you came across the hemopoietic system. Colon, the large intestine, mainly is involved in the absorption of water and soluble vitamins like biotin and excretion of feces by defecation reflex through rectum. The following are the secretions. The saliva secretes a salivary gland, which is 1 liter. The gastric juice is around 2 liters. The pancreatic secretion from the pancreas, which is alkaline, which is 1.2, whereas the gastric juice is acidic in the presence of HCl. Bile, which helps in emulsification, is around 700 ml. And finally, the 92% is absorbed in the small intestine. The succus centricus, which is the small intestine, is around 2 liters. 6 to 7% is absorbed in the large intestine. And finally, the ingestion of secretion and absorption. 1% of feces, water and feces. So finally you ingest and secrete, and the absorption is subtracted. The secretory functions of GIT are: the saliva secretes amylase, which helps in digestion of the starch. So digestion starts in the carbohydrate digestion starts in the mouth by secreting saliva. The gastric juice secretes pepsinogen, which enhances protein digestion. The pancreatic juice or the amylase, the proteolytic enzymes like trypsinogen, chymotrypsinogen, procarboxypeptidase, lipase, cholesterol esterase, ribonuclease, the intestinal nucleus. The predominant is the enterokinase, the maltose, which lyses the maltase enzyme, maltose, into the end products of carbohydrate, the lactase, the sucrase, the dipeptidase, aminopeptidase, and finally the alkaline mucus secretion, which helps in lubrication and protective function. So to understand the concept of gastrointestinal, it is very easy. A carbohydrate which is polysaccharide is broken down into disaccharide and finally a monosaccharide. So the end product of carbohydrate metabolism is glucose. Similarly, in a protein digestion, the polypeptides are into peptides and peptones and finally amino acids. And fatty acids are the triglycerides and the glycerol. For all this, we need some carbohydrate splitting enzymes which are amylase in the mouth and the pancreas, the proteolytic enzymes which is involved in protein digestion which starts from the stomach, and finally the lipase which is the lipolytic which helps in fat digestion. The cross section of intestinal wall is it is made up of serosa, the outer layer, the longitudinal muscle from outer to inner and explaining your the circular muscle, the submucosa, the mucosa. Gastrointestinal tract is a smooth muscle which is 200 to 500 micrometer long and 2 to 10 micrometer in diameter, arranged in bundles containing around 1000 parallel fibers. Each muscle fiber functions as a syncytium because of gap junctions that allow sodium ions to pass through one fiber to another. A few connections exist between longitudinal and circular muscle. This is the outline. The outer is the serosal layer with the longitudinal followed by the circular, the submucosal and the mucosal layer which is lined by the epithelium. Under the mucosa we have lamina propria and the muscularis mucosa. And you can see the two nerve plexus which is called enteric plexus. The submucosal plexus is Auerbach's and the myenteric plexus which is between the circular, the muscle layers. What are the types of glands in gastrointestinal sac? Special types of complex glands like acinar glands with ducts. The unit of a gland is acini. In salivary gland, we have three types: the parotid, submaxillary, and submandibular, which is the serous and the mucous type, and the exocrine pancreatic glands. The tubular glands, which is present in the stomach and duodenum. In duodenum, we call them as villi, the functional unit, and the gastric glands, which secrete HCl, intrinsic factor of Castle, and pepsinogen. The brunus in the duodenum secretes an alkaline mucus. The invagination of epithelium into submucosa. In small intestine, it's called as crypts of Libacon. The types of cells which are present are the epithelial cells, which are the enzymes and electrolytes. The goblet cells secrete mucus. The Paneth cells secrete enzymes. The argentaphane cells secrete serotonin. Special types of gland like liver secrete bile salts and lecithin for fat digestion. Bile salts help in emulsification of fat. How is the secretion? What is the mechanism of secretion? All the secretions are mediated by neural reflexes. These are called as gastrointestinal reflexes, and it can be also hormonal. The psychic reflexes which are conditioned are mainly concerned with the cephalic phase of secretion through parasympathetic nervous system. This mainly is due to the sight of the smell, the sight of the food, the smell, taste, sound and thought of the food. So whenever you see or smell or taste, there is some secretions and all the gastrointestinal secretions. The higher centers like cortex, medulla, hypothalamus, along with dorsal nucleus of vagus, salivary secretions, gastric juice and vagovagal reflexes, which are mainly due to the mechanical distension. And the chemicals which are involved in stimulation of the receptors are sensory, afferent and vagus, and finally the secretion increases due to parasympathetic stimulation. There are also local enteric reflexes which are mainly which are elicited due to distension, and there are some chemicals which stimulate the receptors and which in turn stimulate the enteric nervous system, and thereby the secretion happens. So to understand the most of the time the mechanism of secretion is due to either neural path or chemical, that is the hormonal. The neural is all the reflexes, and chemical it can be due to all the gastrointestinal hormones. The hormonal control of secretion is, there is gastrin, which there are two types of gastrin, G17 and G34, which stimulates HCl secretion. The CCK-PZ, which is the cholecystokinin, stimulates secretion of enzyme-rich pancreatic juice from the acinar cells. The secretin, which is the first hormone to be discovered by Baylis and Starling, stimulates secretion of bicarbonate and watery pancreatic juice from ductal cells. Gastric inhibitory peptide inhibit gastric secretion. VIP, also called as vasointestinal polypeptide, stimulate intestinal secretion and inhibit gastric secretion. What is the nerve supply of gastrointestinal tract? Innervation is by the local path, which is the enteric nervous system, which we explained the myenteric and the Auerbach's plexus in structure. And there is also autonomic nervous system, which is the extrinsic nerve, which is the enteric nervous system. It lies in the wall of the muscular layer and below the submucous. So it's made up of two plexus. The outer myenteric or Auerbach's plexus lies between the longitudinal and circular muscle and controls gastric motility. The submucous and the meiscners, which is lying beneath the submucosal plexus, controls the gastrointestinal secretion, blood flow and motility of muscularis mucosa. This is the general structure, which is from outer to inner: the serosal layer, the muscular layer, the submucosa and the mucosal layer, which has the enteric nervous system, which is the myenteric, which is also called as Auerbach's plexus, and the submucosal, which is the meiscner's plexus. The myenteric plexus, when stimulated, the effects are: it increases the tone of the gut wall, it increases the intensity of rhythmic contractions, increases the rate of rhythmic contractions, also the speed of conduction of excitatory waves along the gut wall. Thereby the food moves aborally. The inhibitory nerves in myenteric cause relaxation of the muscle and decrease in tone. The sympathetic nervous system, which is a part of the autonomic nervous system, relaxes the gut wall, whereas the parasympathetic relaxes the sphincters, and the vasoactive intestinal peptide may be helpful for receptive relaxation in the stomach. The neurotransmitters involved in the enteric nervous system may be excitatory or inhibitory. So E means excitatory, I means inhibitory. So acetylcholine is predominantly excitatory along with serotonin, and so most of them can have dual actions, inhibitory and excitatory. The eccentric control of nervous system, it is made up of parasympathetic and sympathetic. The preganglionic parasympathetic, that is from the craniocercal outflow, supplies the esophagus, stomach, pancreas, first half of colon, very little to small intestine, and sacral plexus, which is the pelvic nerves with S2, 3, 4 supplying the distal half of the colon. This is the nerve supply of the vagus and the pelvic nerve. Whereas the postganglionic parasympathetic, which is located in the myenteric and submucosal plexus, stimulate the secretion, which can be salivary, gastric, pancreatic, all the secretions, and it also stimulates contraction of the gas wall, resulting in peristalsis and relaxes the sphincters. The sympathetic is mainly from thoracic fifth segment to lumbar second of the intermediolateral arm of the preganglionic nerve, and the postganglionic, which is the celiac and the mesenteric. The neurotransmitter released by postganglionic fibers is noradrenaline. Adrenaline is released by adrenal medulla, whose cells are modified ganglia. The sympathetic eventually relaxes the gut wall and contracts the sphincters. The preganglionic fibers of both sympathetic and parasympathetic release acetylcholine, which binds to nicotinic acetylcholine receptors of the ganglia, whereas the postganglionic fibers of the parasympathetic release acetylcholine as neurotransmitter, which binds to the muscarinic. So there are two types, nicotinic and muscarinic. Autonomic ganglia are cell bodies of postganglionic fibers, which are the celiac and mesenteric. Which are the fibers which carry the information of the GIT? They are called as afferent fibers. The sensory nerve endings are on the gut epithelium or gut wall. They can make connections with the plexus of enteric nervous system like the prevertebral ganglia, and they ascend up towards the nucleus of vagus and other parts of CNS. Afferents travel along with sympathetic nerve or vagi. 80% of vagus nerve are afferent and initiate many vagal reflex signals returning to the GI tract through vagal afferents. There are some gastrointestinal reflexes, the colocolic, gastrocolic. The enteric nervous system and its connection with parasympathetic and sympathetic brings about three types of gastrointestinal reflexes. Reflexes that occur entirely within the enteric nervous system, reflexes from the gut to prevertebral sympathetic and back to the gut, reflexes from gut to spinal cord or brainstem and back to GIT. The reflexes that occur entirely with enteric nervous system are the peristalsis, which enhances the movement of food from oral to the anus end, which is based on the law of gut. The mixing contraction, which helps in mixing of the secretions. The local inhibitory effects, stimulation of secretion and inhibition of secretion. Reflexes from gut to prevertebral sympathetic and back to the gut. Enterogastric reflex, which signals from the small intestine and colon and inhibit the stomach motility and secretion. Colono-ileal reflex signals from the colon that inhibit ileal empty, and pain reflexes that inhibit gastrointestinal motility. Reflexes from gut to spinal cord or brainstem and back to, from the stomach and back to gut to control motility and secretion. Gastrocolic reflex signals from stomach to cause evacuation of colon through parasympathetic, and defecation reflex, that is what passage of motion is mainly by signals from the rectum that travel to spinal cord and back to produce powerful colonic and rectal and abdominal contraction of defecation through parasympathetic and somatic afferents. How is the GI motility controlled? It's mainly by neural and hormonal mechanism. The neural, as already know, there are two types, the enteric nervous system and the autonomic nervous system. Gastrointestinal reflexes control motility. As we came across the resting membrane potential, the action potential in the muscles, GI smooth muscle, which is involuntary, have slow waves and spikes, and this is conduction. It's a graded potential. There are four types of membrane potential changes: the slow wave, the spikes, The pacemaker potential, which is the basal electrical rhythm, changes in voltage depolarization or hyperpolarization. The slow waves are the basic electrical rhythm. Most gastric intestinal contractions are rhythmic. Rhythm is determined by frequency of slow waves called basic electrical rhythm, which is present in the fundus of the stomach. It's caused by undulation of sodium potassium pump activity, resulting in a rise and fall of resting membrane potential. Intensity or amplitude of the waves are 5 to 15 millivolts. The frequency of basal electrical rhythm varies at different sites of GIT. And the body of the stomach it's around 3 per minute. In the duodenum it's around 12 per minute. In the terminal ileum it is 8 to 9 per minute. BER does not usually directly cause muscle contraction but controls appearance of spike potential or action potential, which leads to muscle contraction. The spike potential caused by depolarization. The smooth muscle has an unstable resting membrane potential and obeys the property of plasticity. When the resting membrane potential increases minus 60 to minus 50 to 60, the threshold rapid change in RMP occurs known as action potential. The frequency is about 1 to 10 per second. Duration of spike potential is about 10 to 20 milliseconds. Depolarization is caused by entry of calcium and sodium through slow sodium calcium channels. Entry of calcium leads to initiation of muscle contraction. There are pacemaker potentials. Is acetylcholine increases amplitude of spike and duration of plateau. Norepinephrine decreases amplitude, and these are the neurotransmitters which increase or decrease the activity. Change in the voltage of RMP can be due to stretch of the muscle, stimulation of acetylcholine, stimulation of parasympathetic, and stimulation of gastrointestinal. The temperature also can regulate the change in depolarization. Tonic contraction of GIT is not associated with basal electrical rhythm. It is caused by repetitive waves and by hormones, to some extent substance causing entry of calcium. Hyperpolarization is RMP is decreased and the muscle is less excitable, whereas in depolarization it is excitable, it may reach the action potential. The hyperpolarization is mainly induced by epinephrine, norepinephrine, gastrointestinal hormones and other neurotransmitters. As we discussed, the hormonal control of gastric motility can be the gastrin, which can cause contraction of stomach muscle or relax the pyloric sphincter. The cholecystokinin inhibits stomach motility, causes contraction of gallbladder, contraction of pyloric sphincter, and stimulates intestinal motility. The GIP, also called as gastric inhibitory peptide, inhibits stomach motility. VIP inhibits motility. Secretion inhibits stomach and small intestinal motility. Motilin initiates migratory motor complex, which happens during hunger contractions. Digestion. So the major steps in GIT are ingestion, digestion, absorption, and egestion. So we have come to the second part, that is the digestion, which is the basic chemical process of digestion, which is hydrolysis or cleaving by addition of water molecule. The end products of digestion is already I discussed. The glucose from carbohydrates, the amino acids from proteins, the fatty acids and 2-monoglyceride from fats. Absorption is the end product of the three metabolisms, is by three mechanisms. It is mainly by active transport, primary active transport because of sodium and calcium, secondary active co-transport in the presence of sodium, glucose, galactose and amino acids, bile salts in it. Diffusion can be facilitated diffusion for fructose and simple diffusion of water through There is also called a solvent drag, which is mainly assisted by glucose. Fatty acids and cholesterol are re-esterified, complexed to lipoproteins and enter in the lacteals as chylomicrons. Thank you.